Welcome back. We are still in the midst of discussing how biophotonic technology could be utilized to detect genetic disorders. And in the previous class, a long class, we discussed about what genes are and how genes can create proteins, and these proteins creates us. This is what we are. In today's lecture, we are going to understand about the biosensing. You need to understand the disorder, and now you need to understand the sensor part. So, today we are going to discuss the sensing mechanism a bit, and after these two classes are over, we will go directly on to the application of how technology can be utilized to detect disorder. So, welcome again. Today we will discuss the biosensing background. So, let us start. What exactly is a biosensor? So, the 10th World Congress on Biosensor decided that biosensor is an analytical device incorporating a biological material either associated with or integrated with a transducer, and the transducer can be optical, electrochemical, piezoelectric or anything. So, let us break it down. A biosensor, for all intent and purpose, is a transducer. It is a transducer, meaning its property changes upon contact with a specific material. That material is called the analyte, and that material is something that we want to detect. A transducer is a machine, is a material which upon contact with another material modifies its property. The modification of its property, if it is an optical material, then changing refractive index. If it is an electrochemical material, then change its conductivity. If it is piezoelectricity, same, its piezoelectric effect, micromechanical, mechanical vibration, it will change upon interaction with this analyte. This analyte is what you want to detect, what you want to sense. So, the combination of this analyte with transducers creates this biosensor. Now, here comes the crux of the material. Crux of the point is that the transducer has to have a very particular and very specific response, meaning its property will modify, to a particular unit, the change in refractive index, change in conductivity, change in mechanical vibration will have a specific value with a specific analyte only. It cannot randomly fluctuate when any ABCD XYZ material comes in contact with it. The modification in the property of the transducer has to be very specific to particular materials, particular analytes. Analytes are the material that you want to sense. It could be a virus, you want to detect a virus. It could be some kind of a hormone like a pregnancy hormone, you want to detect whether a person is pregnant or not. Blood glucose detection, you want to detect the glucose from the blood. So, the idea is all these machines, all these transducers, it could be the home pregnancy detection kit, or it could be the blood sugar detection or the pulse oximeter. It will show a particular response only when the blood oxygen or blood glucose or the pregnancy hormone is in contact with it. It cannot simply change your blood has so many different materials, right, not just oxygen. So, your pulse oximeter will have no value if it detects, if it starts changing its value by detecting atmospheric oxygen. Or your blood glucose, you just put some amount of sugar onto the material and it spikes up. It has to have a specific reaction, a specific modification upon interacting with analyte. There is a display, a computer screen, an LCD display. In home pregnancy detection kit, there is a color change which results from when the property has changed, when the it's like the display of a multimeter, right. The resistance have changed, the electric current has changed, the conductivity has changed, but you still need a value, you still need to see it in a display. So, that is the display, and this most important part, this biorecognition unit, this determines that a specific analyte is producing a specific reaction, a specific modification. We will be dealing a little bit more with this biorecognition unit. Biorecognition unit allows a specific analyte to attach with a particular transducer, resulting in a particular change in its property, which is then displayed and you are able to see. So, biosensors are analytical devices that can detect chemical or biological species or a microorganism like viruses or bacteria. They can be used to detect toxins, bacteria and viruses both in vivo and in vitro. Yeah, in vitro I told you vitruvius, in glass i.e. petri dish, in vivo, life, so inside living organism. So, main application clinical diagnostic, drug development, environmental monitoring, food quality control. So, there are so many plethora of applications of biosensors. We are mostly interested in optical biosensors because this is a photonics class at the end of the day. So, what happens in most cases you have electromagnetically active structures that have a particular optical response when they have an incident light on top of them, right. They have a particular diffractive index, they have a particular optical response, they reflect a specific amount of light when they are excited with a particular light. You can consider it as fluorophore, fluorochrome, etc. When this electromagnetically active structure is covered with analyte, right, the observed signal changes from its native signal. A fluorophore was emitting red light upon excitement by green light. Now, this fluorophore is attached to something else. Now, that red light has become infrared light. Usually, the energy loses because previously the light was traveling from air to that material. Now, that light has to travel through the analyte, refractive index higher, it has to do more work, loss of energy results in wavelength shift. So, the overall electromagnetic active structures, the original one was optical fiber, then ring resonators, waveguide, metallic nanostructures, those are plasmonic structures, photonic crystals, 2D materials, all of those things have started coming up, meaning they have a particular response of their own. Light passes through them, bounces off like in optical fiber or ring resonator, plasmonic structure, a native response. You cover the material with some sort of an analyte, there is a change in refractive index resulting in redshift in the observed signal, which in turn can be corroborated with the amount or the type of analyte present, right. Different type of analyte will give different type of observed signal, so on and so forth. It will go on and you detect it, you biosense it. So, optical biosensor utilize a change in amplitude, intensity, phase, frequency or polarization of light created by a recognition element in response to physiological change for the direct presence of the analyte, chemical or biological. So, without analyte, this is the native response, lambda versus intensity, right. When the analyte is present, when you have made the blue curve, there is a shift either in frequency or in the phase, even the polarization can change, or the intensity of light that usually used to get transmitted or used to get reflected is somehow getting lost because of the presence of an extra material on top of the optical biosensor. So, this is something that you detect. The how much this lambda will shift with respect to change in refractive index. Previously, the refractive index was 1, now you have put an analyte with a refractive index say 1.4, 1.5. So, it is 1.4 minus 1, that is 0.4 or 0.5, and delta lambda is this shift, gives rise to the sensitivity. The major changes that we expect are a fluorescence. As I told you, previously the fluorochrome was emitting red light upon excited by blue light. Now, the same blue light is making it emit infrared light. Why? Because the fluorochrome is now in contact with another material which you are trying to detect. Absorption. it was the optical material was absorbing at a particular frequency. Now the absorption has increased or decreased or have shifted to a different wavelength. And scattering, Raman scattering, of course, I do not need to explain Raman scattering, we have discussed this pretty well. It was scattering a particular photon, maybe the energy of the photon was at a particular frequency compared to the incoming photon, higher or lower, Stokes or anti-Stokes. Now a completely different photon is being scattered, because now it is not simply that material which is scattering the photon, it is that material plus the analyte which is scattering the photon, and the result there is a Raman shift, Raman scattering modification, change has happened. So, let us come to this biorecognition part. How exactly we specify? So, the crux of the material of any sensor, biosensor including, depends on this specificity. How do you ascertain that your pulse oximeter is only detecting oxygen, or your home pregnancy detection kit will only detect those pregnancy hormones and not other hormones? How do you know that your blood glucose meter is only detecting glucose, not anything else in your blood, and giving false information? You have to be very, very specific. So, enhancement in sensitivity and selectivity, the selectivity is the most important part of the optical response, is achieved by immobilizing the biorecognition element on the electromagnetic active structure. What are these biorecognition element? Enzymes, binds with a specific reactant substrates and catalyzes its conversion into a product. Enzymes are very, very specific. Any chemist, ask any chemist, enzymes are very, very specific. They will react only with certain substrates and create a product. With this product, you can attach the particular analyte. If this product is not present, the analyte will not get attached. These enzymes create this product, and thereby it helps you to attach with this particular analyte. We have antibodies. Antibodies are very, very selective. We will see in a moment for selective binding to antigens. Aptamers are oligonucleotides or peptide molecules that bind to specific target molecules. DNA or PNA, this is not RNA, this is PNA, peptide nucleic acid. I told you DNA has a particular strand. So, AAAT strand will only attach with TTTA, nothing else will attach, nothing else will attach. So, supposedly you are trying to understand the other complementary form of DNA, a particular DNA which results in particular strand. You just give a single strand of DNA with its complement. It will attach or it will not attach. That will help you detection. Very, very specific. So, let us go into this antibody and DNA based biorecognition. This is fascinating. Antibodies, those immunoglobulin, IgG, they are these Y-shaped proteins. I showed you these Y-shaped proteins in previous class, right? Consider this as a key. This is part of your immunity. This is part of your immunity response. These antibodies attacks different types of antigens. Antigens are these pathogens. Antigens are part of pathogens. You can say it's a paratope, but don't let me confuse you. Antigen for the time being is part of a pathogen. It's slightly complicated. I don't want to say anything wrong, because an antigen can also be a pathogen under certain circumstances. But for the time being, think you have the coronavirus, right? You have seen images of coronavirus. Those spikes could be considered for the time being the antigen, and the entire virus could be considered as protein. Those spikes could be considered as antigen, the entire virus is pathogen. Now, they enter your body. Their first response is this antibody. This antibody goes and attacks it with this antigen binding site, this antigen binding site. I will give you another example. Consider the antigen as a lock and your antibody as a key, right? Almost 90% of all locks and all keys are similar. The fundamental difference from one particular key to another particular key is the groove. This groove attaches with the opposite complementary groove inside the lock and opens the lock or closes it, i.e. modifies its property. The key will not get anywhere else into the lock. The key will have a particular place where it will be inserted inside the lock, and if the groove of the key matches with the complementary groove inside the lock, it will open or it will close. No other groove, no other key will match, will open it. So, a particular key is specific particular to a lock. A particular key is particular to the lock. But if you look at any key or any lock, they look very similar to one another. They look very similar to one another. The difference is inside the groove, inside the threading of the key. Yes, that is the only difference, and that makes a particular lock for a particular key. Quite similarly, your antibody makes these types of antigen binding sites, these antigen binding site that need to complement, that needs to attach with specific regions of the antigen, capture it, lock it and change its property. The number of these immunoglobulin are quite common, few 4, 5 Ig, M, IgG immunoglobulin, but these antigen binding sites are as different as the amount of pathogens or as different amount of antigens are there. This part is common, this part is very, very specific, and just like a particular key attaches to a particular part of the lock, these antigen binding site of your antibody attaches to a particular virus, particular pathogen, particular bacteria only, and prevents it from hijacking the cellular mechanism of your body and cause any harm. The immunity of a person is determined by how quickly or how nicely your antibodies, this immunoglobulin, are able to determine, are able to create a specific antigen binding site upon invasion of a particular antigen. If your body fails to make this, if your body fails to make the key that will unlock the lock, you are in trouble. You are in trouble. It takes certain amount of time for your body to develop that immunity, to develop that key, develop that groove of the key. If your body succeeds, you win literally in life. If your body fails, your immunity fails, then this antigen takes over your body, this pathogen takes over your body. We can externally help our immunity by different processes, vaccines, medicine, exercise, etc. But at the end of the day, your body has to create a particular binding site with respect to a specific antigen. So, the same thing goes with DNA based biorecognition. You put fixed probes with a particular set. If it is complementary, it will attach. If it is non-complementary, some of them will attach, some of them will not. Partially complementary, this is fully complementary. You then send it to some sort of a sonicator. Remember the nanotechnology class, wash it. Since they are covalently bonded, they are much stronger. Since they are hydrogen bonded or less covalently bonded as compared to this will flow away, this will remain. This is then recognized, this is then looked into change in its optical response, and then we put antigen antibody complex at certain areas of our body. You can immobilize your analyte using adsorption, spin coating, or via covalent bonding. The requirement for a biosensor is it is able to develop multiple analytes simultaneously. It can create lab on chip platform, it can have IoT and communication biosensing. So, the first thing that they did was trying to work on optical fibers. Optical fibers, as you know, you have a core versus cladding, higher refractive index, lower refractive index, and thereby you have total internal reflection. Optical fiber, I do not want to go too much detail onto this, but for non-medical people, optical fiber is something that gives you the internet through which you are able to see me, where light is completely confined inside the core. Light simply bounces back from one end to another end and passes in a total reflection mechanism. So, what we do is that we put specific antibodies at specific end of the optical fiber, body of the core or the top of the core or the tip of the core, specific antibodies for specific antigens, they will get attached. Previously, this light was having a particular frequency, it was passing through the core and coming outside into air. Now, it will pass through this and come and interact with this specific antigens, and thereby there will be a change in its response. Change in the refractive index results in modification of the output light. The modification of the output light is determined by the presence or absence of a specific antigen connecting with a specific antibody. You can either put it at the top of or the tip of the core or at the body of the core. Similarly, you can have structures such as this, functionalized MNF sensing, multiple areas, multiple network formation. You can have lambda 1. If there had been no antigen antibody, then you had have got lambda 1 itself, but now you have modification, you have lambda 2 with and without analytic. You can have optical fiber sensors. There are several other type of sensors, maybe at a next class we will discuss a bit about them, but for the time being optical fiber I am telling you because they are the oldest and pretty common these days. Optical fiber based soil sensor, optical fiber based water sensor, you have used optical fiber in endoscopy, blood biopsy, distributed sensors, so all of those things are there. You have blood flow, in which Doppler flowmetry has been used. This is another fascinating thing. Light is being sent through several of these small cores into a vein. Into your vein you insert this, light is being sent and some of the light is being reflected, and since there is a flow, so this is a vein having blood flow and there is a blood clot. You know about Doppler shift. When things move away from it, from you have a red shift into the frequency. So, think about it. You are standing at the junction in a road. A car is coming, blowing its horn. The frequency of the horn is same for the car, right? But if the car is far away from you, you hear a different frequency. When the car comes, you hear a different frequency, and when the car goes away, you hear a different frequency, right? Try this. It is not the intensity, I am not talking about the intensity of the sound, it is the tone, it is the frequency of the sound that is different. Doppler effect is exactly that. Doppler proved that as a body moves away from us, as a body approaches us or moves away from us, the frequency changes. There will be a blue shift if the body is coming towards us, there will be a red shift into the frequency if the body is moving away from us. This is widely utilized in astronomy to see the red shift, the Doppler shift of a particular star, particular galaxy vis-a-vis our planet, meaning the universe is expanding, the stars and galaxies are going far and far away. We try to utilize the same principle in blood vessels into your veins. You have put your optical fiber tube somewhere, they are very small, few micron, they can get inserted into your blood veins, etc., and try to see if there is no blockage, the red blood cells will completely flow away from it. It will come near it and it will flow. You are standing in the road, the car is coming and it will passing through. So, there will be a blue shift and then there will be a red shift, but if there is a blood clot, there will be no shift for a long period of time, right. Where it is scattered and partially absorbed by it, light scattered by moving blood cells suffer from a Doppler shift, which is analyzed by the detector and is processed to derive the blood flow rate. As it goes through, you have a Doppler shift, a red or blue shift depending on the movement. You can understand the blood flow rate, and if the blood flow rate is different from normal, you can understand that there will be a blood clot. Fascinating. So, this is some sort of a biosensor that utilizes optical information Doppler shift to detect if you have a blood clot or not. So, these are the various concepts that I covered today for biosensing. Very briefly we discussed biosensing, and from next class onwards we will go into full detail onto how biophotonic technologies can be utilized for detection of genetic disorder. By this time you have some rudimentary idea of gene, you have some rudimentary idea of biosensors. From next class onwards we combine both of them, sensors to detect genetic disorders, sensors to detect genetic materials to determine genetic disorders. So, these are my references and I will see you in next class. Thank you very much.